Okay, so
this is not, perfect. Yes.
we should be past the testing phase by now.
Yes, yes, I think so. So I guess without further ado, it is my great pleasure to welcome Caroline Uhler. And she is an associate professor at MIT in the Department of Electrical Engineering and Computer Science. Carolina started out as a biologist, actually, and I'm hoping that she's going to spend a few seconds telling us about her path, and then trained as in mathematics at the University of Zurich, and then ended up doing a PhD in statistics at UC Berkeley. And she joined MIT in the Simons Institute, the program at the Simons Institute from UC Berkeley, and had various postdoctoral positions in various places around the world, including ETH Zurich and IST in Austria. She has, her work has attracted many accolades and awards, and I guess I will mention, for example, the NSF Career Award and the Simon Investigator Award, and so on. And so it is a very great pleasure to have you here, Carolina. And she works on the, I guess, intersection of machine learning, statistics, and computational biology. So not a million miles away from what we've heard from Marinka. So I suspect that there might be some synergies there. And so it's my great pleasure to welcome you, Carolina, to tell us about causality in particular and autoencoders. Thank you very much.
Thank you so much for this introduction. And yes, let me start off with a couple of words about my path, as was already introduced a bit here. Yeah, so I'm Swiss, so I went to the university in Zurich. But actually, what maybe put my path like even earlier on was that I really love teaching. So after high school, I taught for 1 year. I taught all kinds of different things. I taught Latin, I taught German, French, math, all in secondary school. And even before that, my aunt was a teacher, so I often taught for her when she was not available. And so I really started university wanting to become a high school teacher. And how I chose what I wanted, what I actually went to university to learn is just what I enjoy teaching the most. So to me, that was math. So I started off in math, was always also interested in biology. So in the end, wanted to actually teach both. So I did a master's in math and, a bachelor in biology at the same time. And also did a master's in education. So in high school education. So in Switzerland, you actually have to get a master's in, whatever the subjects are like math and biology in order to become a high school teacher in those areas. And so it was only in the last year, in fact, during university that I started thinking about doing a PhD. I never even thought about it before, didn't really know what it meant and what you would do in a PhD. And so that's, and why that happened is actually my PhD advisor, who became afterwards my PhD advisor at UC Berkeley. He was at ETH Zurich giving a course. He was there on sabbatical and he gave a course and this course was called Algebraic Statistics for Computational Biology. And so during my undergrad, I actually always studied statistics, algebraic geometry, and then biology. And I had no idea that these things could be combined in any way. To me, they were all separate. And, I chose statistics because I saw that as a way to kind of connect math and biology as something that is that actually allows you to look at data. So that's how I got to statistics. And algebraic geometry, I just like from the thinking of my brain works more geometric. And so that was kind of the area I really enjoyed. And so he was offering this course and I got actually from something like 4 people or so independently who actually based on their advisors, PhD students, based on their advisors had to take this course. I got all of them emailed me that this would be, this is exactly a course made for you. And so this is how I learned about this course and actually then went to this course, which is called Algebraic Statistics for Computational Biology. And I loved it, right? It was like one way of actually connecting algebraic geometry and statistics and biology all in one. Yeah, and then I decided to do a PhD based on that course that I've taken. And so this is actually kind of a funny story. I contacted my then, to-be PhD advisor, Bernd Sturmfels. And I asked him whether I could meet with him and whether I could do a PhD with him. And, I knew nothing about PhDs in the US or anywhere, in fact. But I did learn by then, like how it works in Switzerland, that you just, ask an advisor whether he would take you, right, as a PhD student. And so he rightfully asked me then, do you know anything about the PhD system in the US? And I was like, no, I have no idea. And he was like, well, I'll be happy to work with you if you get into UC Berkeley. And I was like, okay, so what does that mean? So what do I have to do, right, in order to get into UC Berkeley? And so, okay, so then I figured out, I have to apply. And I was like, well, I don't want to have to TA the whole time, although I love teaching, right? But actually not being like, just having my own funding would be kind of nice so that I can decide whatever I want to do once I'm there, right? And just be independent. It felt kind of odd that like some advisor would be like the person who will either have to pay me and yeah, that I just don't have my independence back. I guess I always had my independence during university. I was always teaching and had my own money, etc. So coming from that background, it was kind of odd. So that's when I looked into fellowships and, I just Googled, right, fellowships US. I hadn't, I mean, my family, there was nobody who did a PhD or went to university or anything. And I also didn't have this background at the University of Zurich where, anyone could really help me or would help, or, maybe I just didn't know who to ask. And so I Googled, and then I heard about, I found out about Fulbright. I had never heard of it before. And I was like, okay, so let me apply for a Fulbright fellowship. So then I actually got a Fulbright fellowship. That helped hugely because they then did the whole application process. So I could give them like a list of 5 universities with this Fulbright Science and Technology Award, which would cover me for 3 years, and then I could give them a list of 5 universities where I would be interested in applying to, and then they did the whole application. I just had to write like some essays, etc. So that was super helpful, and that's and then I got into Berkeley, and I was like, well, I'm in Berkeley, so then Bernd was he was very, I was like, okay, so I'm here, so can I now work with you? So this was really exciting, and I had a really, he's just super supportive, and particular for women in math. I think he has played such an important role in like actually educating and taking so many female PhD students. And PhD students, in fact, with all kinds of backgrounds. So he has really been a huge supporter and a really great mentor. And yeah, so that has brought me to Berkeley, where I was then in the stats program, PhD program, and was always still interested in biology and different kinds of applications. With him, did mainly algebraic statistics. So, how could statistical models be written as algebraic varieties, and how can you then use different insights from algebraic geometry to actually get better, say, figure out what it means for estimation, for parameter estimation, etc. in statistics. And was on the side, because I had this fellowship, right, was completely free to do whatever, was also always continuing to work on biological problems. In particular, I was sitting in Lior Pochter's lab, and was also doing things on phylogenetics. So usually did one semester on more biological questions and one semester on more mathematical questions. And then again, the last year of my PhD was kind of an interesting one where I had to decide what I wanted to do. So I basically had like 1 year where I was like going through all different kinds of jobs and trying to figure out whether, any of them would be something that I would love to do. In the end, I think I was like, so I went, so I talked to all kinds of also industries, etc. And in the end, I was like, well, maybe scientific consulting is something that I would be excited to do. But then also realize that, even also if I'm a faculty member and I still thought I would really enjoy teaching, even as a faculty member, I could still do things like scientific consulting, right? So in some sense, I could do everything that I felt like I understood I enjoyed by then, after taking a lot of time thinking. And so then it was actually very helpful once I really decided that, okay, I want to become a faculty member, just having gone through that process, because once you know what you want, it's so much easier to then actually put in the time that, you need to spend in order to actually apply for these jobs. Okay, so then came, of course, a problem with Fulbright that, you have a 2-year home residency requirement. So I wasn't even really allowed to apply for any faculty positions in the US. So then I was concentrating on Europe, and in Europe it is of course difficult to get a faculty position just after your PhD. So there was basically only one place I applied to, and that is IST Austria. They were, just starting off having like more of an American kind of system where they would actually take people with just a PhD, and I was very excited that I actually got an offer from IST Austria and then moved there as a faculty member. I did take 1 year off, I guess. I did half a year. I went then for half a year to ETH Zurich, and for half a year, I took half a year off. I went for some months to ETH Zurich and for some months to the Institute of Mathematics and its Applications in Minneapolis for a program. And IST Austria has been great for me at the beginning, just having time in a research institute, having time where you don't really have to teach as much, and it's very small, and just having time to really figure out what your research agenda is. I think just after a PhD, at least for me, it did take some time to really set it all up and have time to just start off small with like postdocs, having some guaranteed funding, being able to slowly apply for all kinds of grants as well. But in particular, mainly working with postdocs was, I think, very helpful. And I did that for about 3 years and then moved to MIT in 2015. And have been super excited at MIT. So here I'm in electrical engineering and computer science at the Institute for Data Systems and Society. And so it was really exciting to finally have a lot of students to work with, and just like a lot of people around who are actually doing, who are interested in similar kinds of problems, and just having a lot of people around to actually discuss science with, who are just super excited to discuss any, research at this intersection, either on the statistical side or on the mathematical side or on the computing side or, on the biological side. And now most recently have started as the co-director of the newly launched Eric and Wendy Schmidt Center at the Broad Institute. So that's a new center that is really thinking about how to not only bring machine learning into the biological sciences, but also go the other way around. Of what we really want to do is actually try to think how we could change what is currently being done in machine learning by actually motivating it more from problems in the biomedical sciences. So we think that, biomedical problems are really quite different, or can be quite different of what they require from ML than your more standard applications like online advertisement and online education and self-driving cars and recommender systems, etc. And in particular, that they actually often require getting at underlying mechanisms instead of just prediction. And Marinka said it at the end as well, right? You often need interpretability, and in particular, you need mechanisms. And so that really requires something a bit different from ML than your standard applications. And so what we're trying to do is really make the biomedical sciences one of the main, one of the goal applications that you start thinking about when you're actually developing and trying to think of ML problems. So that's where we are today. I'm really excited about this interface and really excited about, thinking through what are the big questions at this interface between the biomedical sciences and machine learning, and really grateful, to Eric and Wendy Schmidt, who actually are funding this, and the Broad Institute who are and the Broad Foundation who are funding this endeavor, and, really excited to actually get this going since now a couple of months. Okay, so that was a little bit of an overview of where I'm coming from and how I got to where I am today. And with that, let me actually give you one little story of this, of how we're thinking about particular, how biological questions can really then lead you to actually developing new theory on the deep learning side. And this example is actually coming and very related to what was just presented before on COVID-19 and drug discovery. And I will take it a little bit more into the theory of autoencoders and theory of overparameterization, etc., where there has been a lot of development recently on the machine learning side. And just to show you that actually these theoretical questions are really necessary in order to get to important biological applications afterwards. Okay. So let me get started with drug developments against COVID-19. So here, I don't have to say as much because of an amazing talk that you just heard. So I'll here take it more into the drug repurposing direction. Although the method I'm going to propose is nothing, it's not just applicable to drug repurposing, but I think in particular in the COVID-19 context, at least at the beginning, we were thinking that, since it is so urgent, maybe we want to concentrate on drug repurposing. So looking at drugs that are already FDA approved, and that could then be quicker in a much faster way, actually taken, through trials, because they don't have to go through the safety trials anymore. They have already passed these and could just directly go to the efficacy trials to actually be able to test whether, this drug will also be successful in a new context, or effective, I should say, in a new context. Okay, so in terms of computational methods, so you've seen now a lot about, graphs and drug discovery based on graphs or protein interaction networks, and you can use different kinds of graphs. So here I'll mainly be talking about actually a different approach. I'll use graphs at the end. But let me first talk about a different kind of approach and tell you about why I think causality is actually super important for both of these, for all of the computational approaches, but is really not yet used yet. So one approach that people have, and both of these have been used successfully for actually identifying drugs from a computational perspective, although most methods are still completely experimental. So one approach that people have been thinking about and have used is the following, where if you can figure out what the disease signature is, right? So that's a vector in space that goes from normal state to disease state. And often, diseases have, mainly affect a particular tissue or a particular organ. So let's look at COVID-19, for example. So they're the first cells that you would be looking at is lung epithelial cells, right? So say you have lung epithelial cells and you look at normal versus diseased state. And you can look at this in different ways. You can look at it at protein data and imaging data and in expression data, whatever it is that you're looking at, right? You can define a disease signature. Can be based on images or expression. In this talk, we do a lot on images, but in this talk, I'm going to do it based on expression. So that's a disease signature, okay, which goes from the mean of normal to the mean of diseased. And now what you want to do is find a drug whose signature is pointing the other direction, okay? Very, very simple, kind of, this is known as signature matching in drug discovery literature. So you're just trying to find a drug that actually points you in the other direction. And you can just, you can list or rank drugs based on how anti-correlated they are with the disease signature. Now, there is important things to note here, and this is what I'm going to do in this talk, is that the drugs can have very different effects on different tissues, so on different cell types. So, it will be important that what we're doing is looking at the disease in lung epithelial cells, and now we need to find a drug that in this context will be pointing back in, is most anti-correlated with the disease signature. So that will be one approach, and this is in fact an approach that I will be taking mainly in this talk, and talking about, what are the difficulties about it from a machine learning perspective, etc. Another approach, and you've now heard a lot about drugs, about graphs, is if we're looking at, and this is actually very heavily used, is if we're looking at these, for example, protein-protein interaction networks, and again, protein-protein interaction networks are not disease specific, and they're also not cell type specific. So often what you do is you need to then figure out which part of this huge graph to actually look at. So often you add like node values, and you've seen this now, for example, differential expression for this particular disease, right? So that you know where to concentrate on in this graph, and then say like here I have a particular part of the subnetwork that I'm looking at, and then one particular standard approach is to look at nodes that are most central for it, and look at these proteins could be maybe good targets for drugs. Now there is a problem with this, and here is what I want to suggest in terms of like why causality actually matters. And there's not a problem, it's just maybe causality might actually help you a lot to filter out targets that will definitely not work. So, underlying this network, although the network that we can get out of, if you look at protein-protein interaction network, is just whether two proteins actually bind. And so these, that's why it's not cell type specific. You just take two proteins and you just look at whether they bind, and then there is an edge there. And it's, you never know whether these proteins even appear in the same cell type, right? Even if this cell even has these two proteins. So that's why this is not cell type specific at all. But, anyways, underlying this network, there is actually a causal network, because all of this is a regulatory network of whether, whether a particular gene will actually turn on or turn off another gene. And then, you have a protein that comes out, which is actually performing its function. But underlying it, there is always some causal regulatory network. So if the regulatory network, something looks like this, right, where everything is pointing into this protein that you're targeting, well, then this protein has no effect whatsoever, right? Because, it is actually regulated by the disease-associated genes and not the other way around. So really directions matter, right? So you do need to take a causal approach in order to at least find proteins or subnetworks that look like this, where, if you now target this node, then at least it has the possibility of getting these disease-associated genes back to the normal state. There is no guarantee, right? It might actually be doing the wrong thing of like making it even more extreme, but at least the edges are pointing in the right directions that it could have a regulatory effect on these other nodes around. So this is just to show how causality will actually be really important, no matter which approach you take. And I'll talk about this first approach also to tell you why this is in fact also causality enters into this approach here, where we're trying to do the signature matching, which is, the main part of this talk that I'm going to talk about. Okay, so let's talk about the signature matching approach. And so here we're getting more into, what the data actually is and how does this maybe relate to anything that you have seen in machine learning. Okay, so these are the standard data sets that we actually have available, and they're freely available for anyone to use. This is this known CMAP data set. You also have the same on imaging side, but this is the one that I'm going to use. So this is expression data where you have, thousands of different perturbations. So these are different drugs, but all kinds of other perturbations as well, genetic perturbations, etc. So let's just think of them as drugs. Okay, so many drugs that were applied to many different cell types. Now the cell types here, this particular data set, and in fact, this is the largest one that is available, freely available, was done for studying cancer. So all of these different cell types here, they're actually cancer cell lines. Okay, so that's what we have, and every entry here that is black, what you get is a 1,000 dimensional vector. So this is a 1,000 dimensional expression vector. So humans have 20,000 genes, so usually it's a 20,000 dimensional vector. Here, to just reduce the costs, right? This is a huge experiment. They only measured 1,000 important genes in humans. So that's what this looks like. So this is a tensor that is only partially filled in, right? So only these black entries are the ones that you have. All of these here are free. And whenever you have an entry, right, you'll have the whole row in terms of you get to measure the whole 1,000 dimensional vector. Okay, so this is the data that we have. And so these are all of the different perturbations. You have many of them applied to some cell types, right? And for some cell types, you only have very few perturbations applied to them. And so from this data, what you would like to do is, well, the effect of drugs on some cell types, right? Oops. The effect of drugs on some cell types. Well, can you now from here predict the effect of these drugs on SARS-CoV-2 infected lung epithelial cells? That's really what we want to do, because then, if we can predict the effect of these drugs on lung epithelial cells, SARS-CoV-2 infected lung epithelial cells, then we can just look at, which one is most anti-correlated with the disease signature, right? And just rank the drugs based on that. But as I said, drugs don't have the same effect on different kinds of cell types. So just because they have this effect on this particular cancer cell line will not mean that they have the same effect on a SARS-CoV-2 infected lung epithelial cell. And that is well studied and well understood in the pharma industry. So how do we go about it? So you see here already, if you take your ML hat, this is probably some kind of matrix completion problem, right? And I'll take it maybe a little bit slightly different, but we do have now matrix completion view of this as well. But I do want to point out this is actually not your standard matrix completion problem. Okay, so this is not your standard like Netflix kind of thing where you have maybe here movies and here people that are rating the movies, right? And then you want to fill in these entries, because this is actually a causal task. So when you have a movie and a rater, it's more like you're trying to predict a conditional distribution, right? I'm conditioning on this movie, and now, you know, what will be the rater, what will be the ratings? Whereas here, a perturbation is actually it's an intervention, right? So a drug or a genetic intervention will only have an effect downstream of it. So this is what is known in causality. It's a different operation. It's not conditioning. It's not marginalization. It's known as this do operator. So, you know, it's something different, right? Because it will only have an effect on nodes that are coming downstream of it in the causal ordering. So it is known that it can be the same as marginalization. It can be the same as conditioning, but in general, you know, it is like on different nodes, it will do different kinds of things. So this is a very different operation, and it's not the same as your standard matrix completion problem. It is actually a causal. I mean, you can say it's a matrix completion problem, but it's a causal matrix completion problem. So your standard approaches generally don't work well. Also here, I should say that anything low rank will not work well, right? Because if as I said, you know, if one of these entries is missing, the whole row is missing. So so that means that if you do anything low rank, it's just going to fill in a constant, and that's certainly not a good completion in this particular framework. So anything like low rank will actually not work in this particular framework. So what is an approach that, you know, another approach that you may want to take if you are putting on your ML hat, and is another, you know, very intuitive approach. And this is, you know, we're now going to go in this direction. Happy to also talk about the matrix completion one. We do have a paper on it online as well. So so this is a different approach that people, you know, might be thinking about in this setting, is like the style transfer approach. Okay, so we could see a drug as a style and be like, okay, so I know what the drug does in one context, which is in the context of a cancer cell line, right? And now the question is, well, what will this drug actually do in the different context? So can I just transfer it over somehow from one context to the other? Okay, so how does this work generally, right? So, and I'll be looking at autoencoders because we do have theory and understanding of autoencoders, although often, you know, GANs are used for this kind of application. But you'll see now soon how actually theory of autoencoders will be really, really helpful when we're trying to come up with a good framework for actually solving this problem in the causal setting. Okay, so so here we have, you know, say you have all kinds of images of people. Some are in neutral face, some are in smiley face. There are some people who just never smile, right? And you would like to produce the image of this person here smiling. Well, what has turned out to work quite well is GANs or autoencoders used for this, where, you know, you first train your autoencoder on all of the images that you have. So that means you're getting this latent representation. And now for some people, you do have a neutral and a smiley face. So then that means that this vector here in the latent space corresponds to adding a smile. And so now actually taking this vector and just moving it around, in particular, you can move it to a person who doesn't smile, right? And now just add this vector, out comes a point, and I can decode it back into my original data space. In this case, it's images. And in fact, out comes a person smiling. This particular person smiling, right? So this has been quite effective, in fact, in all kinds of vision applications, and in fact, in all kinds of other applications as well. And so it is maybe intuitive to try to use something like this for drugs, right? So you do know the effect of a drug for one cell type. So that's this. Well, let me just take this vector in the latent space, right? And just move it over to the cell type where I do want to predict what this drug will actually do. And, you know, maybe outcomes the drug, the cell type with this drug applied to it. And maybe this actually works out. Okay, so this has actually been done with your standard autoencoders in this paper here for just a couple of perturbations and just a couple of cell types. And it has worked quite well. So the question is, of course, does this work more generally? And, you know, if you talk to people in AI and in the pharma industry, they have tried this. And unfortunately, you know, it can work, but it often, in fact, does not work. And it is maybe not surprising that generally this will not work. And you know, even what we're going to propose, you know, generally, so it will work because, you know, we have some theoretical insights of when it will actually work. But generally, you should not expect this to work. There is, in fact, work in causality by Barenboim and co-authors, in particular, you know, various papers written with Barenboim and Pearl on this is known as causal transportability. Okay, so, and maybe more known in the policy literature, where say I have a particular intervention or policy that was introduced in, say, you know, London, and now I would like to know, well, whether what will this policy do if I will also introduce it in Boston? And you know, and this is known as causal transportability. Okay, so I have an intervention in one setting. Well, what will this particular intervention do in another setting? And can I actually predict what it will do? Even just this, like, you know, even just whether it is predictable or not. So this is the question of causal transportability. And you know, Barnboim and Pearl, they actually gave necessary and sufficient conditions for causal transportability. Now, it is not applicable here because their necessary and sufficient conditions assume that you know the full causal structure. So you know exactly what the underlying regulatory mechanisms are of how this policy affects, you know, all of the covariates that you're measuring. You know exactly which ones of the nodes are the ones that are changing between London and Boston. And you know exactly where the policy, which nodes are affected by the policy. If you know everything, like the full causal structure, then there is a criterion which is necessary and sufficient, which is a graphical criterion. So all of this is like, we have no assumptions on the distribution, which is really nice. It's completely non-parametric. So it is a graphical criterion just on the network, which tells you whether, you know, it will be possible to actually find some formula, which will tell you what the effect, how it will change, right? How this policy will affect if you introduce it in Boston. So that means that, you know, these necessary and sufficient conditions are pretty strong. So definitely it's not always possible. And in fact, you know, they tell us that usually it is not possible, but we don't know the causal graph, right? So we cannot apply any of this. All it says is that it's certainly, you shouldn't expect it to always work because there are necessary and sufficient conditions for when this will work in theory. Okay, so, and in fact, we can just test it out, right? We can apply these autoencoders. Here we have this wonderful data set where we have thousands of different perturbations, and some of them, actually many of them, have been applied to multiple cell types, right? So let's just test it out. Let's just see in the latent space whether the effect of drug 1 on cell type 1 is aligned with the effect of drug 1 on cell type 2. The effect of the drug should be pointing, you know, should be pointing in the same direction, right? Because what I'm going to do is just take this vector and move it over. So if the vectors are not aligned, well, then there is no chance of getting anything here to work. So let's just test it out, right? Let's just test it out using our autoencoders, and we actually see that in general this doesn't work. And you know, we're not the first ones. So I know from working with people in the pharma industry, they have done this before and they have seen that this doesn't work. So let's actually just do it. And so this is what I'll be doing. So this is your standard autoencoder, where, you know, you go from your large, so autoencoders are standardly used for dimension reduction. You go from your high dimensional space to a low dimensional space back to your high dimensional space, right? It's always seen as like this non-linear version of PCA. And now here comes our crazy, and this is where all the theory will come in, and I'll motivate why we're even doing something this crazy. So we'll do something crazy. Okay, so we'll look at we'll use autoencoders for embedding them into a higher dimensional space. So we'll go from, you know, our whatever 1000 dimensional space to a higher dimensional latent space, and then back to the 1000 dimensional space. And I'll motivate this, but, you know, nobody has ever used these autoencoders before because it doesn't make sense, because, you know, you have more parameters here than you have at the beginning. So you have enough parameters to just learn the identity map, right? There is no constraint, there is no bottleneck anymore. So we definitely have enough parameters to just learn the identity map. Now I'm actually showing you already on this picture here, and I'll go through it, that the autoencoders, if you train them, they're actually not going to learn the identity map, and they're going to learn an interesting map. And then I'll give you the theory of what exactly they're learning. Okay, so, but this is your standard autoencoder. This is what has been used for these kinds of applications. And I'm going to show you what actually happens when you're using an overparameterized autoencoder. And what you should get from this page is that maybe, you know, it is doing something interesting, and it's certainly not learning the identity map. Okay, so let's look at this. So what am I even plotting here? So here what I took is just 2 cell types. Okay, namely, so here I took 2 cell types that actually have the most drugs applied to them, so that, you know, we have most of the entries actually filled in, so that we can just look at them, whether now the drugs are aligned or not aligned. And we did it for all drugs over here, and I'm not going through it, but, you know, just believe me, it's exactly the same. If you want to do it for all filled in entries, you can also do that here. Okay, and so now what are we doing? Well, we're just looking at how much aligned, right, based on correlation, how much aligned are the signatures of the drug in the original space. So if you would not do any autoencoder, and then in your lower dimensional embedding, and we have done it for different kinds of embedding dimensionalities here. Here I'm showing it to you for 100-dimensional embedding. Believe me that this is the same whether you do it a little bit more or a little bit less. Okay, so this is just going to a lower dimensional latent space and looking at the correlations in there. And what you see here is that you basically get everything along the diagonal, but you do get a little bit of an enrichment up here, which is great, right? These are the drugs where it actually works, where they're aligned and you can actually, where you have causal transportability, right? Where you can actually move between them. But you see that most drugs actually just fall on the diagonal, meaning that nothing is happening. So it doesn't really matter whether you're doing it in the input space or in the lower dimensional embedding space. It's just basically doing the same. Of course, in terms of, you're using a bottleneck. So in terms of reconstruction, you do have some loss, right? You have some information that was lost. Okay, so now what happens when you use these overparameterized autoencoders? And in fact, what we can prove, and we have proven, is that in fact, the more overparameterized you are, so here you see that I'm not super overparameterized, right? I'm going from 911 to 1024, but you can go higher and higher, and we can prove that this thing here is actually going to be a step function. Okay, that actually all of the drugs will come out to be aligned. And so, but what is really important to note here is, first of all, it's doing something interesting for this alignment problem. And second of all, it's certainly not learning the identity map, right? So this was learning much more something closely to the identity map than this one here is, or, at least it's learning something along the diagonal. It might not be the identity map at all. But here, right, it's for sure not learning the identity map because it's learning something completely different, right, in the original space as compared to the overparameterized latent space. And of course, in terms of what have you lost by doing this? Well, nothing, because we're overparameterized, so we have training error 0. So we did not lose any information whatsoever, but we did not learn the identity map, right? So we are learning something that seems interesting for this particular application. Now you could ask me, well, what about PCA, right? That's the other way of doing it. Well, let me just reduce it to two principal components. I can also do more, and we did over here more. So what happens then? Well, what is actually quite interesting is these two pictures look quite similar, right? When you're just going to two principal components. So actually just using the first two principal components also allows you to align drug signatures across different contexts. But the problem is that you're doing that by just throwing away basically all the information in the data. So another way of getting alignment is just by throwing out basically all of the information, right? But that's probably not how you want to get alignment to work, because in the end, you still needed to do well on your drug prediction problem in the end, right? So you definitely don't want to achieve it just by throwing it away in all the information, but that's what you see here, right? So you can basically not reconstruct anything. So you can, but you can get, there are different ways of getting alignment, but I'm just saying this is probably not the one that you want to be using. Okay, so this is just to show that maybe these overparameterized autoencoders are doing something interesting. Just a little bit of intuition for why even look at them. For us, it came from the theoretical insights that I'll show you. But just at a very high level, if you want vectors, like if I go back to this picture, if you want vectors across contexts to be aligned, well, if you go to something lower dimensional, you're actually crumbling up the space more. So it is much more difficult to have things be aligned in a lower dimensional space than it is in a very high dimensional space, right? If I'm giving you more and more dimensions, it's much easier to keep things aligned that were aligned, or actually to make things aligned. This is just at a very high level. Of course, this is no proof or anything, but this is at least a little bit of intuition why large dimensional latent space should help you if you need things to be aligned as compared to low dimensional spaces where you're crumbling up the space even more because you have actually very little space, right? All of the information that you had in your 1000 dimensional space suddenly has to fit into your 100 dimensional space. And obviously then a lot of things will be crumbled up and become much, much more nonlinear than they were before. So that's just a little bit of intuition. There is no intuition yet for why this autoencoder actually doesn't just learn the identity map, right? Because it can, because you're here, you have no bottleneck. So I'm now going to go into the theory for overparameterization and what are actually these maps that are learned by these overparameterized autoencoders. Okay, but that was just motivation for why we should look at them. So here, just very briefly, some theory for it. So this is like your standard autoencoder, right? Now I'm going to the overparameterized setting. I'm going to assume, so overparameterized for me just means that you have training error 0. So you have enough parameters to exactly interpolate the training data. So this is your standard, map of the autoencoder applied to a training data set, a training example minus your training example. So you're just trying to minimize the reconstruction error. Okay, so now I just want to give you a little bit of intuition for what might actually be happening here. So let's look at the simplest setting where we just have one training example and our autoencoder is just a linear map. Okay, so then we would have something like, right, a times x minus x. And we're trying to minimize this. I can do 2 norm and, you know, I'm trying to minimize this. A is where in R k times k, right? And x is just a vector in RK. Let me write times 1, right? So this is then the problem that I'm just trying to solve when my autoencoder is just linear and I just have one training example. So that's a very extreme case, but it's actually quite good in terms of just getting at some intuition. So if I try to minimize this, then it's clear that there are many solutions, right? So first of all, the minimum is always 0. So I can definitely find a matrix A so that this minimum is 0. Now the question is, what is the argument? So what is this matrix A that is learned? Well, there are many different solutions, right? So it could, for example, be the identity map. So A equals to the identity would actually solve this problem. But there are many other solutions. It could be, in particular, it could be the projection onto, you know, a rank 1 solution, right? Which is just a projection onto the sample. That's another solution. Or it could be a rank 2 or rank 3, et cetera, right? Solutions to this problem. So the question is, what is actually the autoencoder learning? So which ones of these solutions out of the many, right? It is going to get training error 0. So which one is the one that the autoencoder through gradient descent, or, you know, you're going to use stochastic gradient descent, et cetera, is actually learning? So for the theoretic, so of course, you know, our autoencoders are trained in a standard way, but for when we try to prove something, all of the results that you're going to see is always analyzed when we're using gradient descent. And, you know, as is standardly done, our autoencoders will be initialized, so all of the weights will be initialized close to 0. So this is exactly the theoretical setting that we're looking at. So all of the proofs will be gradient descent initialized as close as possible to 0, so that you can still, you know, move away from 0. So that's the theoretical kinds of setting. But, you know, our standard, just to show you how these things actually work and get a little bit more intuition in it, here is just a result using a standard, you know, unit autoencoder trained on one example. And I should go back in the theory. So here, this is well known. Okay, so if we just have, if this is the problem you're trying to solve using gradient descent initialized at 0, what you're converging to is a rank 1 solution. This is well known. The question is, how does this actually generalize when we have non-linearities and when we have multiple training examples? Okay, so that's not known, and that's what we will be proving. So here is just to get some intuition for what is actually happening. So here is your standard unit autoencoder trained on one training example. Okay, and so now I want to look at what is this map that is being learned. Well, how do I actually test the map that is being learned? I just feed in some random images, and here, you know, let me feed in some nice cars or turtles or even noise or, you know, anything you like. And what you see is outcome always these cute little bunnies. Okay, so what the autoencoder actually learned is a point map, so much more extreme than a rank 1 solution, right? A rank 1 solution could also have given me the negative of the bunnies. Here you see that what the autoencoder actually learned is a point map onto these bunnies. Okay, so some people might say, well, this is what I would have expected. So let me show you that maybe this is not what one should expect, or it is actually quite surprising because there are infinitely many solutions, right, that the autoencoder could have learned. In particular, let me show you that so a unit autoencoder on one training example is actually quite deep. So in particular, let me show you that if you actually go very shallow autoencoder, in fact, it looks completely different. Okay, so for a shallow autoencoder, what you're actually going to learn is something completely different. It looks much more like the identity map. So here you see, you know, whatever we're putting in, well, something quite similar to it comes out. Now I'll prove on the next slides, and you know, this is, I think, important to note because there are papers that say that these autoencoders actually learn the identity map, that this is not the identity map. Okay, it's provably not the identity map, and although it might look like very close to the identity map, I'll show you on the next slides that it is not the identity map. And here is what happens in the linear setting. I'm actually showing it for 2 training examples, and this is very easy to prove that, you know, what comes out is a linear combination of your training examples. Okay, so so here you see it quite cleanly that it is always a linear combination of your training examples, but of course you can get the positive and the negative, as I said before. So it is quite surprising that in this setting here that we saw, where it's quite deep and you have one training example, you actually get out the point map. Okay, because it has to do with the nonlinearity, and it definitely has to do with depth, as you see here. So if you don't have nonlinearity, you get linear combination of your training examples. If you don't have depth, you can actually get something that is quite close to the identity map. Okay, so now how do we analyze what is actually the map that is learned? So what we're going to do, and it's such a simple idea, but it will go through with all the proofs, etc., is, well, if you want to understand whether this is the identity map or not, well, the nice thing about an autoencoder is it goes from, RD or RK to RK, right? So I can just iterate the map and see what happens. In particular, if this is the identity map, then even if I put back in this horse, I should just remain in the horse, right? If I iterate it. If it's not the identity map, well, then something interesting might happen. And that's exactly what happens. And so let me show you what actually happens when you iterate this map. So here we trained it on, say, 500 training examples. And, in order to identify what this map does, instead of just putting in a random example, which we also did, here I'm showing you examples where we took training examples and we just added a whole lot of noise to it. Well, what is interesting, if you actually iterate this map with a whole lot of noise added to it, in the first iteration, you would get out something that looks like the same. Okay, so that's not even what I'm showing you here. So if you iterate multiple times, then, you get to something like this, etc. You keep iterating. What is quite interesting is what you end up with is actually one of your training examples. And it's also quite interesting to analyze what are the basins of attraction, right? How much noise can you add in order to end up to the correct training example? Or when do you end up in the wrong training examples? And what can we prove about it? Can we, for example, prove that all our training examples are going to be attractors, etc., etc.? So, but what this already hints at is that, this is a nice way of interrogating what are the maps that autoencoders are actually learning. And it suggests that what these autoencoders, these overparameterized autoencoders are actually learning are maps that are contractive at the training examples. And here, is just written this out on a 2D example where we just made it up, where we just, have here our training examples in 2D, and we just iterate the map on a grid just to show you how the basins of attraction look like. And you see that, these basins of attraction are actually quite interesting, right? And they actually depend quite heavily on the nonlinearity that you're using. And so this is a completely open problem that we don't know yet, but will be very important to understand is how these basins of attraction actually look like that you're actually learning when you're using these overparameterized autoencoders. So in terms of overparameterized autoencoders, so you can actually make quite small, there's always a question of how large does my network have to be in order to be able to memorize a whole lot of images. Here, for example, we trained an autoencoder. You don't need a lot of width or depth in order to actually be able to memorize, say, 500 ImageNet examples. And make them all be all of the training examples are attractors. You can prove that they're all attractors, right, by just looking at the eigenvalues of your Jacobian in these training examples and show that the largest eigenvalue in absolute value is smaller than 1. So that's how we can prove that, in fact, this autoencoder has all of our training examples be attractors. Now, what is our theorem? And so this is a theorem that appeared in PNAS. We now do have the general theorem. So this is for one training example, that we can make one training example be an attractor if you just have depth and width be large enough. We now have the proof that you can make any number of training examples be attractors. And we also have implemented that if you just make your depth and width be large enough. So this result here that you identified here, we actually do have a proof that is in fact, that we can also prove that that's the case. So, but how does this proof work like? So here we're just looking at one training example. You take some autoencoder, depth D and width K, some nonlinearity, initialization close to 0 is where we can prove that this usually happens. Here it says suitable conditions on the nonlinearity. So all of the nonlinearities that are standardly used fall into this, satisfying the suitable conditions. So these are very, very relaxed conditions. And so what the theorem says is that, it's actually it's just a formula for what the maximum eigenvalue of the Jacobian actually is for a given nonlinearity and for a given depth and width. And so now, all we proved in here is that, well, once we have this formula is that, now you can actually choose your depth and width large enough so that your training example will become an attractor, just by that you can actually make this eigenvalue be smaller than 1. So it's a very descriptive kind of theorem that actually tells you that, how you can make this particular training example be an attractor. Maybe a couple of remarks on this, because these are questions that I don't always get. So one question is about generalization, and one question is always about I'm using memorization in a very particular way, and in fact not equivalent to interpolation. And I would like to point that out because I think it is kind of important, and often it is used interchangeably. So there are many maps that can interpolate training data, and this is being described like very early on, of course. So here, if I have an autoencoder, a very simple one that goes from R1 to R1, okay, and I have some training examples, then any function, right, that passes through these training examples interpolates the training data. Now, I will claim that, a function like this definitely does not memorize the training data, because there is no way of you. So to me, memorization means not just interpolation, but actually being able to recall the training examples. Well, there is no way for you from a function like this to be able to recall the training examples, right? Because how do I know that this is a training example, but not this dot over here or this dot here? So just because it interpolates doesn't mean that you can actually memorize the training examples. Whereas if we're proving that the maps that we learned are attractive at the training examples, well, that's a way for actually getting back your training examples, right? Because you just iterate. So this is a kind of map that we prove that you're learning, right? Then if you just iterate this map, well, then what comes out is, is the step function, which is just a step function at your training examples. So this is what we showed happens with strong depth, right? Just empirically. So we don't have a proof yet of this. So we showed that if you have like very high depth, then what happens is just basically you just get a point map out at your training example. So that empirically is quite clean. If you don't have a large depth, then what happens is that you get this function that is contractive at the training examples, that if you iterate it many times, then you're going to get out this function here. So that's the difference between interpolation and memorization that I think is kind of important to make, that we don't use them interchangeably. The other question is usually about generalization. So, does memorization, like that we prove here that it memorizes training example, does that mean that it doesn't generalize? So I think, first of all, we should define what does actually generalization mean for autoencoders. I have not seen a good definition yet. I don't think we have had enough discussions about it. Sometimes it is used as, it learns the identity map, that means generalization. I'm definitely not agreeing with that definition, because if you really want to learn the identity map, then don't train anything. Just I don't need to train in order to learn the identity map. So that certainly doesn't make sense to me, at least as a definition of what it means to generalize well. So another definition could be, for example, there should be debate about it, and I don't know, but maybe this work suggests that another definition of generalization could be a map that is as close as possible to the identity map, but does, where all your training examples are attractors, right? Because you definitely want that if some test example comes out to be close to a training example, it should not move away from the training example. It should remain close to that training example. So you don't want the function that I have here on the left, where you can be very close to a training example, but you're just moving infinitely far away from it. That's probably not a map that you may want to consider in terms of actually generalizing well. So this could be a definition, and I think there needs to be a lot of debate about it. This is in fact what we're using, what seems to work well on our biological applications, just motivated by this, is in fact, I have, so again, here we don't have proofs about it, about, what is the effect of width and depth. I'll just say it in words and then, show you that we do have some empirical results, but this is how we're now always using our autoencoders is, so depth, as you saw, right? So what depth actually does, and you can analyze it on these, I've looked at these plots so many times that I kind of see trends, but probably by looking at it the first time, one doesn't see the trends as cleanly. But what happens if you increase depth is your eigenvalues move more towards 0. Okay, so they become much, much more attractive, right, at the training example until it is a point map. Whereas if you just increase width, it's the variance of your training examples becomes smaller. Again, we don't have proofs for this. This is all empirical. So I think it would be really important to actually prove these things. So now what we always do is we have our width as large as you want, right? And now, in fact, we're always looking at infinite width networks as well. So the neural tangent kernel regime. But okay, so you let your width go to infinity, meaning, all of your eigenvalues will concentrate. And then we just have enough depth so that you have all of your eigenvalues be smaller than 1, so that you become attractive at your training examples, but you are, still as close as possible to the identity map. This seems to work really well for the kinds of biological questions we're working on. It's motivated by the results that we have here, but is motivated by our, definition of what we will propose here, what it means to generalize well, where, I think there should be a lot of discussion about, but at least it generalizes well in the kinds of applications that we're working on. So that's the intuition that we got out of this. And I know I should stop here, and I am actually just getting back to the original application. So this is how we got to these overparameterized autoencoders. Now, from these overparameterized autoencoders, you get the signature of a drug out, right? So the signature of the drug, now I can just look at which one is most anti-correlated with the disease signature. I actually get out a ranking of all of the drugs. Now here, these are FDA approved drugs that we got out. A lot of them are cancer drugs. You may not want to use them, in fact, in the COVID-19 context, just because they have so many drug targets, and they of course have a whole lot of side effects. So what we looked at then is like, well, maybe there is in fact a drug that you could develop that is where, now we know the targets, right, which we believe are the important targets. Maybe you can actually now, if you know the right target, maybe then you can develop a new drug that is more specific to this particular target, as compared to these cancer drugs here that actually have many, many different targets. So if you can figure out which ones of these drug targets is a causal one, then you may be able to just identify a particular or develop a particular drug just for that particular target. And so we try to just hone in, now use actually more our standard, methods for learning directed graphs to figure out which one of these drug targets is most upstream to all of the disease associated genes. As I showed at the beginning, our network, we would like to figure out which ones of these drugs here, which one of these targets or genes is actually most upstream, right? Actually, at least pointing out towards all the disease-associated genes. And, there is actually only one target, which is this gene RIPK1, that always turns out to be most upstream of all of the disease-associated genes. So we have, over the years, spent a lot of time on developing methods for learning causal graphs. So we actually learned these causal graphs and try to figure out which one of the targets is most upstream. It's quite interesting that this RIPK1 actually directly binds to SARS-CoV-2 proteins. It was not something that we put into our network. So this is the target we identified here. What is also really exciting to us is that actually this an inhibitor for RIPK1 just entered, different phases of clinical trials for COVID-19, and is being developed by Sanofi, and is still, continuing on its ramp in the clinical trials. So it has been quite exciting to see that this might actually be something interesting. Is also something where we actually understand what the mechanism could be. So here, actually working with biologists to really understand what the mechanism is of how RIPK1 might be acting. I think it's always really important that we do work together, so that, it's not just computational methods, but that you actually then understand why is something coming out, and does it actually make any sense whatsoever. And so that's this RIPK1, which maybe I don't have to go into here. But with that, let me just end and conclude with some remarks. So I hope I was able to show that, a principal causal framework is really critical for drug discovery, whether you're doing it at the network side of things that I only did at the end, or whether you're looking at it from this matrix completion problem point of view. It is a causal problem. It is predicting the effect of a perturbation. So we do need to take causality into account. So also showed how, it is important to then get to the theory of machine learning, where, you do need to understand what these neural networks are doing that you're using. And you may, through that understanding, actually be able to develop new types of frameworks that might be actually particularly important for the applications that you're looking at, but also maybe more generally applicable to many different kinds of applications. Autoencoders to us have not only been an extremely useful tool in biology, but also generally about studying in neural networks what it means to be overparameterized, and understanding what are the functions that these overparameterized neural networks might actually be learning, and why they may be useful. They show remarkable self-regularization schemes, as we have already seen for neural networks, overparameterized neural networks used in the standard classification setting, right, where you've seen like this double descent kind of work by Misha Belkin and afterwards by many others. But here, what we see is that these overparameterized neural networks, they learn contractive functions. What is quite interesting is that Benjo, for example, had very early on regularizers that he added in to make autoencoders contractive at the training examples. So here what we showed is that just overparameterization itself will do the same trick. You don't need to add any regularizers. And the more overparameterized you are, the more self-regularizing these maps become. So they just do it by themselves. It's this really nice property that you can also achieve with regularizers. If you really, that was shown that you can add regularizers to achieve this. You just use overparameterization, and this is exactly what it's going to be doing. Okay, a lot of open problems. As I already mentioned, we really need to understand these metrics that are being learned by an overparameterized autoencoder. Seems to depend on a nonlinearity that you're using, but we need to understand this. What are these basins of attractions that are actually coming out of when you're training these autoencoders? And with that, let me end and thank, of course, an amazing group of students, in particular on the theoretical side of what I showed you on overparameterization, Adit's work here, together with Misha Belkins. But the SARS-CoV-2 work was really a big lab effort that we all worked together on, in particular also with our collaborator on the biological side, who's been a very long-term collaborator, G.V. Shivashankar's lab at ETH Zurich. And then, of course, nothing would be possible without the amazing funding. And thank you all for your attention.
Thank you very much, Caroline. So we are running a little bit over time, but I think we're going to have time for at least a question or two. So we have one on the floor here in the physical room. I shall just pass the microphone.
Thank you very much. Thank you very much for the very interesting talk. I really enjoyed it. I was just wondering, in your examples, you had the input and the output spaces were Euclidean spaces, so you had a linear structure. But in many applications, you might have just a graph or maybe a Riemannian manifold as input. For example, images can be sometimes represented very well. Well, the distance between the images can be represented as the Wasserstein distance, and then you have some sort of a Riemannian structure. So I was wondering if you were to extend your results to such metric to this metric setting.
Yes, so I think this is a great question. So in some sense, so here we're not taking into account, right? So we're just showing that it is contractive in whatever space. I mean, in the examples, we are doing it on images right here. So for example, so we are doing it on images and showing that as a contractive in the images. And we did look at how convolutional structure interacts with this particular phenomenon of being contractive at the training examples. And convolutional structure, what it does is it requires you to have more depth than if you just use a fully connected network. So there is definitely some interactions there. And I think in terms of theorems, I mean, so we have also done it with graph neural networks. So I think the intuition, like standardly, is, and even with graph neural networks, we're now always doing this overparameterized setting. Just for the biological applications that we're working on, it always seems to work much better. And it is, maybe it's something else, and you're getting at the right questions. I don't have the theorems that you're asking for. I just don't have them. But another thing of why we're using it is that it also usually helps you for training. So it is much, much easier to train these networks. They're much better behaved when you're just overparameterized. So there's two things, right? It seems to generalize better, and it also seems to help a lot for training. But I think the questions you're asking are the right ones. And, I would actually ask the question in the latent space. And that's why I'm putting in these questions here of, what are the metrics that are learned in the latent space? And we really don't know. I just simply don't know.
Thank you so much.
I think great questions. Mm-hmm. Sorry. I don't hear you.
Hello, can you hear me?
Now I hear you. Perfect.
Okay, I probably have a few seconds. I was wondering if your input is a metric space, could your middle space, this big high dimensional space, be a space where you would want to embed your original metric space? Could it be interpreted like that?
Yeah, it would be really nice to understand that better. We really just don't know, but I think those are exactly the right questions. I just don't know the answer. Yep.
Thank you very much.
Yeah, and maybe one more thing, you know, the latent space, these pictures that I have here might be also completely misleading because, right, we're going from low dimensional to high dimensional space. So the metric in this high dimensional space is not something that we can make a picture of. So showing it in 2 dimensions can be very misleading. But you're asking all the right questions. I just don't know the answers. I see Rima has her hand up.
I think that we are, oh, does she? I didn't see, but we are running out of time. Okay, Rima, you can have a
very quick question. Thanks, Colleen, for the nice talk. I was just wondering, you show this property of self-regularization or this contractivity. What happens if you throw in an image that is not in the test set and then do this perturbation? Do you see
you're actually still you're still going to contract to one of your training examples. So yeah, so that's actually harder to prove, right? But if you see it here, so everything, so here, this whole space, so we actually, in this particular examples and in the overparameterized setting, what is hard to prove is that you don't have any other attractors. But that's always what we see here. You don't have any other attractors. So everything will be contracting to one of your training examples. That's an important thing that we should prove at some point, right? But it's much harder to prove than just proving that you have small eigenvalues on your training examples. And that's why I think it's so important to actually learn a good metric, because what we would really like to do is try to find out, add a training example that has a large kind of area of you know, where it attracts, where all of the garbage is going to be attracted to. That's really what you would like to figure out, like, how can I come up with a particular training example that has that properties, which has a large basin around it, and in particular, all this garbage will just be attracted to it. That would be really great if we could do something like that. And yeah, that's a big open problem.
Thank you.
Great. Thank you so much, Caroline, for this very, very inspiring and exciting talk. But we have sadly run out of time. So thank you very much for this really fascinating time. Thank you, Caroline.
Thank you very much.